Hi, I'm Christina Lott, and this is my new forensic analysis method literature review. I chose to review an article from the Journal of Forensic Science called Evaluating the Sustainability of Current Mitochondrial DNA Interpretation Guidelines for Multigenerational Whole Mitochondrial Genome Comparisons. I was pretty excited to find an article about genetics because that's what I'm currently teaching in my 7th grade science classroom. Definitely not in the same capacity as this article. This article went way in depth into some really heavy genetic terminology that I had to kind of do a little bit of research on my own to figure out what some of these things were because I didn't want to just skim over the information. So my first couple slides are going to be some of the background information that I looked up for myself to help better understand the process that the article discuss. So first off, we are going to talk about mitochondrial DNA. Mitochondrial DNA is found in the mitochondria of cells. It's passed on to an offspring from the mother parent. It can be used to trace your ancestry through your mother's lineage. It has a higher copy rate in each cell, and that means it's often recoverable when nuclear DNA is significantly degraded. So because there's so many mitochondria within one cell, it's often easier to recover this type of DNA versus the one copy that's only found in the nucleus of the cell. The Sanger sequencing method of mitochondrial DNA, also known as the chain termination method, it was developed in 1977 by Frederick Sanger and his colleagues, used to determine nucleotide sequence of DNA. It's previously the only method available for forensic casework where samples were obtained from skeletal remains. This method of DNA analysis is widely used, or was widely used in the past, from 1977 into the early 2000s. It does not accurately account for heteroplasmy. Heteroplasmy is the existence of both normal and mutated DNA in a single cell. Heteroplasmies can be used as genetic markers in applications ranging from forensics to cancer diagnosis. Further research is currently being done in the frequencies of heteroplasmies occurring. The research that I read discovered that these mutations occur more frequent than previously thought. The article that I read focused on next generation sequencing for DNA analysis. It was introduced in 2005, used to determine the order of nucleotides in entire genomes or targeted regions of DNA or RNA. Intended to improve the identification of human remains. Could be used to avoid classifying heteroplasmic changes as differences in human identification, which is often something that occurs using the Sanger method. The next generation sequencing method is cost effective. It's much cheaper than the Sanger method, and it is sensitive to minute variances in DNA samples. Scientific Working Group on DNA Analysis Method guidelines are set in place for forensic analysis of DNA. There's 3 guidelines that come back from lab samples. So if a sample is given the term exclusion, it means the samples differ at 2 or more nucleotide positions, and they are excluded as being from the same source. Inconclusive is when samples differed at a single position. The results are considered to be inclusive. You cannot determine whether they are from the same source. And then cannot exclude. When samples share a common DNA base at every nucleotide position, they cannot be excluded as coming from the same maternal lineage. Current guidelines may not be considered sustainable for entire mitochondrial genome comparisons involving distant relatives. Research aims to evaluate the current guidelines in regards to comparisons involving distant relatives, multi-generational analysis, and analysis that includes the entire mitochondrial genome. Some current hypotheses that are coming from research is that sequencing the entire mitochondrial genome introduces additional variation between maternally related pairs than what is observed when sequencing a control region. The number of meiotic events affects the percentage of nucleotide differences observed between two maternally related pairs. Applying these modified recommendations leads to a reduction in the number of false exclusion rates. Physical evidence that's analyzed by this method. This method can be used to analyze highly degraded biological samples. It's ideal when nuclear DNA is too decomposed for lab analysis. Mitochondrial DNA is more easily extracted from samples of old bone, teeth, hair, and other samples where DNA content is low, and it may be hard to extract nuclear DNA. The usefulness of this method in forensic science: in case of fire victims, recovery of old or highly decomposed remains, instances where only teeth or hair are recovered. The next generation sequencing method is faster and more cost efficient, making it a valuable tool for more forensic labs. Personal opinion and evaluation of this method. I believe that the next generation sequencing method is an admissible source of DNA analysis for crime cases because the validity of the test results have been scientifically proven to be accurate. Because of the high occurrences of mutations in mitochondrial DNA and the fact that it traces a group of individuals' maternal lineage, not just solely identifying one individual, I do not feel that it should be used as the only source of conclusive evidence. It should be supported with concurring evidence under most circumstances. An example where mitochondrial DNA tracing would be beneficial is an occurrence of where a known family member was lost in a fire and the family wanted remains to have a proper funeral. The NGS method could be used to identify bones or teeth for the relatives of the victim. Conclusion: as new scientific methods are being developed, their use should be explored with how they can be applied to the field of forensic science. It's important that the field's best practices and guidelines be kept up to date with the help of new technology and new scientific methods. These new technologies and methods should adhere to the same rigorous testing procedures that all scientific methods are subjected to. Scientists must work closely with investigators to keep each other informed about new practices and guidelines so that crimes can be successfully solved and justice can be rightly served. For my resources, I listed the article I chose for my literature review, as well as the supporting resources that I read myself.